Researchers at the National Eye Institute have discovered cellular mechanisms that help the 13-lined ground squirrel survive hibernation. Their findings could be a step to extending storage of human donor tissues, awaiting transplantation and protecting traumatic brain injury patients who undergo induced hypothermia. NEI is part of the National Institutes of Health. The findings were published in the May 3 issue of Cell. During hibernation, the 13-lined ground squirrel endures near freezing temperatures, dramatically slowing its heart rate and respiration. How the squirrel's tissues adapt to the cold and metabolic stress has confounded researchers. A structure in cells known to be vulnerable to cold is the microtubule cytoskeleton. This network of small tubes within a cell provides structural support and acts as a kind of inner-cellular railway system, transporting organelles and molecular complexes vital for a cell's survival. In a series of experiments, the research team led by Wei Li, Ph.D., a senior investigator in the NEI Retinal Neurophysiology section, and Jingxing O, Ph.D., a postdoctoral scientist in Li's lab, compared cells from non-hibernators to cells from the ground squirrel to determine differences in their response to cold. They found that in ground squirrel neurons the microtubule cytoskeleton remains intact while it deteriorates in the neurons of humans and other non-hibernating animals, including rats. To investigate the biological factors supporting the squirrel's cold adaptation, researchers created hibernation in a dish, took cells from a newborn ground squirrel and reprogrammed them to become stem cells, which are undifferentiated cells capable of becoming any type of tissue in the body. Importantly, these lab-made cells, also known as induced pluripotent stem cells (iPSCs), retained the intrinsic cold adaptive characteristics of the adult squirrel's cells, thus providing a type of platform for studying how various kinds of the rodent's cells adapt to the cold. Next, they compared gene expression of stem cell-derived neurons from ground squirrels and humans. Cold exposure revealed distinct differences in the reaction of mitochondria, organelles that provide energy to the cell in the form of adenosine triphosphate (ATP). They found that cold-exposed human stem cell-derived neurons tended to overproduce a byproduct of metabolism known as reactive oxygen species, ROS. The overabundance of ROS in human neurons appeared to cause proteins along the microtubules to oxidize, wreaking havoc with the microtubule structure. By comparison, ground squirrel ROS levels remained relatively low and their microtubules remained intact. Cold exposure also interfered with the human stem cell-derived neuron's ability to dispose of the toxic oxidized proteins via its protein quality control system. Under normal conditions, lysosomes envelop oxidized proteins emitters and digest them via enzymes called proteases, but in the cold-exposed human neurons, the proteases leaked from the lysosomes and digested nearby microtubules. The researchers then treated non-hibernating cells prior to cold exposure with 2 drugs to alter the course of the cold-induced damage. One of the drugs, 15 Bosnia and Herzegovina convertible marks, inhibits the production of ATP, which reduces the production of ROS. The second drug inhibited protease activity.